Hi, I'm Jonny Wray. So I'm not a computer scientist, I'm a computational biologist. My career, my job is applying computational approaches to biological problems. At E-Therapeutics, we're a drug discovery company based in Oxford. Our niche, I guess what we do differently is all of our early stage discovery is in silico, is computational based, whereas obviously the industry is traditionally an experimentally based industry and still is. However, I think there is a change that's going on in the industry. It's certainly getting a lot of hype around the AI terms that we've heard about. But more generally, the rise in the application of data analytics or computational approaches to drug discovery is certainly at the forefront of our industry at the moment, and I think will continue to grow. And what I wanted to go over today was very high level, actually. Drug discovery is quite a big field, of some of the current applications of computational approaches in drug discovery and some potential future expansions, some of which may get a bit science fiction-y and crystal ball gazing. So, before I get into that, maybe some motivations. So, the world population is growing and aging. Some statistics up there about effectively, the change in the rise in the increase of non-communicable diseases. One on the right-hand side, I'll come back to this in a few slides later, specifically around neurodegeneration and dementia. And as we age, the burden on both the individual and on society of dementia is just going to massively increase. And it's an extreme. Where we are right now, literally 100% of drug trials have failed in Alzheimer's. We have nothing to progress or stop progression of that disease, and not even really affect the symptoms. So it's actually a massive issue. Just to give you a very brief overview of some of the issues, I guess, around drug discovery. So this is a pictorial of the drug discovery process. Two gross phases: a preclinical phase where compounds are identified, potential drugs are identified and then tested in animals generally, or in the test tube in dishes first, and then a clinical phase where we test those compounds in humans, and that clinical obviously goes through 3 phases. Just a couple of points I wanted to make here. These numbers and on the times are actually quite optimistic, I would say, but it's not exactly a quick turnaround. Trying new things, you got 15 years potentially for something you try at the beginning to say, is it going to work or not? It's also costly, which gets me into the next slide. So the one in the middle is a slide just giving some numbers on how much does it cost to get a drug approved, and the current best estimate is $2.5 billion for each approved drug. So there's frankly massive realms for improvement in the productivity in our industry. The graph on the left-hand side was from a paper from 2 or 3 years ago, where they looked at the industry productivity over time, and this is a log axis, and the guys who wrote the paper named this Eroon's law, which is Moore's law in reverse. So basically the productivity of our industry has been going down with time, and this is in the face of ever increasing advances in biotechnology of post-genome. We can do so much more now about measuring biological systems and perturbing them, etc., and it's not helping. The graph on the right-hand side is a bit of an analysis for why drugs fail. The vast majority of drugs fail effectively for biological reasons, either safety or efficacy. Somewhat of a tautology, that's effectively what we're testing. But I guess the point I want to make is that most of the time, especially when you get to say phase 3, 2 or 3, most of the drugs fail because they don't do what we expect them to do in humans. We are pretty bad at determining, is a drug going to work in humans? And that's, I think, one of the major areas of potential improvement of productivity in our industry. And I think where computational approaches will actually have an effect in the coming years. So, you know, I was told to think about the future, and when I think about the future of drug discovery or you know, healthcare, I think of this guy. You walk into you know, his room and he instantly tells you what's wrong and fixes you. Now, is that science fiction, or is that actually going to be the future of medicine? I actually believe it's going to be slightly different. So rather than an instant diagnosis and instant treatment, I think we're going to be moving more to ongoing diagnosis and ongoing treatment. So we basically, catching something early is you know, is the key. We all know that you know, the biggest indicator of cancer survival is early diagnosis. Where the field of neurodegeneration work is going is, again, catching it early and coming up with a treatment that can stop disease progression. Basically, late stage symptomatic disease can't be reversed. So if you can stop a neurodegenerative disease from actually progressing, that's going to be the best treatment. This approach works in certain diseases. If any of you have any relatives or yourselves with rheumatoid arthritis, for example, that's eminently treatable these days by disease-halting drugs. Whereas, you know, a decade ago, it was a massively disfiguring disease. However, we can't do this right now. So what are the requirements? So we need an indicator of pre-symptomatic disease based on cheap and simple tests. You know, you're in a bit of a catch-22 here. People go and see the doctor when they're ill. Well, you're too late at that point. When you have symptoms, you know, coming back to the cancer case, the early diagnosis cancers are generally the ones where there's something obvious wrong in a other indication, in another indicator. That then you go to the doctor, something's wrong. It's like, oh, it's cancer. Some of the worst, lung cancer, for example, is because people ignore the cough. And by the time the cough is bad enough, it's too late to treat. So the early indicators of pre-symptomatic disease are going to be critical for this ongoing diagnosis and treatment. Now, you know, that gives rise to the concept of continuous screening of a population. Well, you know, there's a massive issue here that I don't know if people have read about the current sort of meta-analysis that's gone on with breast cancer and prostate cancer screening, which is effectively saying it does more harm than good a lot of the time, and this is because of this false positive rate. You know, a test can be great, 1% false positives. However, most people, if you do general population screening, won't have the disease. Therefore, most people who test positive are going to be false positives. It's a huge issue. So it's a problem that needs solving if we want to go down this. Like, the way it's currently solved is to define, you know, multiple indicators effectively at risk populations so that you can actually reduce that the background rate, so you're actually testing more people that are likely to be positive. The other aspect of this is, okay, now you've identified that someone's in a pre-symptomatic area of disease. What treatment that we have is going to work for that individual? Could be a drug, could be, you know, other types of treatment, but what treatment is going to work for that individual? You may have heard of the concept of personalized medicine. I'll come back to this in a few slides. But the idea behind that is, in the extreme, is that we will all have drugs tailored to our individual makeup. We're all different. We all have a different genetic background. We've had different environmental influences through our lives. By the time you get old enough to have some of these diseases of aging, you're going to be very different than everybody else. Will it ever get to that extreme of everyone has a slightly different drug? I don't know. That might be science fiction. But the way we're going is certainly segmenting disease into smaller and smaller subpopulations. And then another critical one is continued monitoring for disease recurrence and potentially change of treatment. Biology is, by its very nature, robust to perturbations. gets around cancer drugs routinely. It does very smart things, although it's not actively intelligent. Cancer cells will evolve pumps that actually pump out the drug from the inside of the cell out, so they can't have the effect. Things like this, that they happen, and that's why cancer drugs stop working over time. While I was thinking about this, the sort of parallels to centralized versus distributed computing came in my head a little bit. One of the issues of this, if we are all effectively going towards an ongoing diagnosis and treatment regime, we're constantly being tested for pre-symptomatic disease. We're going to have to move from a centralized to a distributed diagnostic and treatment regime. A health system couldn't support all of us getting routinely tested all the time. So we need tests performed routinely by the patient themselves without doctor involvement, ideally. Potentially treatment plans based on those tests, again, are formulated for each individual. Drugs synthesized locally and targeted at specific individuals. People are working on, we heard about 3D printing in the last talk, about printing out 3D printing of specific microfluidic reaction chambers for localized synthesis of small amounts of compounds. And then the last one, personally, this was suggested to me by a colleague. Seems like science fiction to me, but maybe not, that the rise of nanorobotics that could potentially perform biopsies locally on diseased tissue within the cell, and potentially do the analysis and drug delivery also. So why is it hard? Is there a pointer on here? Oh yeah, the big button that says pointer. Apologies for some of these terms. Phenotype is the biological name for behavior, basically. So what we're trying to do with drug discovery is manipulate disease phenotype. Hopefully for the positive, of course. But we have to do this by manipulating things at this level. Drugs are molecular elements. They bind to proteins. There's a gap here. And predicting what manipulations at this level do to things at this level is the key problem in drug discovery, basically. Laboratory techniques are inherently reductionist. You can only manipulate, a handful of things. Where, if it's more than 10 these days, it's surprising. At the same time, however, these are systems of, many thousands of proteins. So the inherently reductionist techniques don't bridge levels well, and they're hampered by combinatorics. However, computational approaches can potentially give insight into some of these issues. So where are we at the current time? So there's lots of areas where computational approaches have been applied, and I just picked some out that I was somewhat familiar with in the area of preclinical discovery. I'm not going to talk about clinical at all here. So the first one is target identification. Target is a protein that if you manipulate it with a drug, it will have a positive effect on disease. This is generally done by, in the computational realm, it's still really a lab technique, dominated by lab techniques, should I say. But people are looking at statistical analysis of gene mutation to disease relationships to drive target ID. The idea behind this is that, and I actually think it's a bit of a dodgy idea, but the idea behind it is that a genetic mutation in a particular protein can be a surrogate for the pharmacological manipulation of that protein. That's a terrible idea personally, but that's what drives the target-driven discovery here. It has had some successes in very limited sets of diseases, so it's not terrible, but for more complex diseases, it's basically not getting anywhere at the moment. Computational chemistry, this is probably the oldest field of computational approaches to drug discovery, started in the 80s, and this is, can you predict and improve how a compound will bind to a protein target? a lot of work going on here, down to the quantum level modeling. A lot of supercomputer work is done here, and I know very little about it. I'm not a chemist, but it's been successful. It promised the world in the 80s, and the world didn't arrive. It's still used. The limitations are now accepted. It went through the typical hype cycle. Chemical optimization. This is a newer one, and I actually quite like this one. When you discover a molecule at the beginning of a drug discovery process, it's basically a starting point for a chemical optimization program where those compounds are manipulated and new compounds are synthesized and made, and you're trying to effectively optimize multiple properties that make a drug. It's basically a multi-objective optimization problem that's done in the lab. There are companies now that are specializing in effectively applying computational multi-objective optimization approaches combined with laboratory approaches. So they go through a sort of design, make, test cycle within the lab to apply those computational approaches to this problem, and it's actually been quite successful. Predictive toxicology, again, reasonable success there. LAS is a company that some of you may know of that's based in Leeds, actually, that have a suite of tools. They've been doing AI before AI's become popular again. A lot of their tools are actually expert system based, others are just traditional machine learning based, but they've actually got FDA approval for some of their for some of their predictive analysis. That the FDA actually allows those predictions to be used in a submission to allow you to go into the clinic. And then predictive ADME. ADME is absorption, distribution, metabolism, and excretion. Basically, what does the body do to the drug when you put it into the body? Systems pharmacology is the field, I guess, and they use sort of traditional compartmental modeling of coupled differential equations, reflecting, trying to model how the drugs will distribute through the body. So potential future things here. Talked to some of my computational chemistry colleagues. They were claiming that actually some of the issues there at the moment aren't computational. They're more limited by data, basically good crystal structures for the proteins that they're actually trying to do the predictions on. That's one person's view that I work with, might not be correct. I talked to one of my system pharmacology experts, colleagues, and again, the feel there was that they're limited by experimental data at the moment. You could make the compartmental modeling more accurate, reduce the compartment sizes, etc., but the data is not available to constrain that modeling. Come back to my area, more biology. Most of the techniques that I mentioned here are statistical, not mechanistic, and I think that is one of the current limitations going forward with the biology, because I mentioned the personalized medicine a couple of slides ago. Most of the techniques, for example, the statistical analysis here are all population based, and we're going to be moving, we have to move in the personalized medicine world away from population based predictions into individual based predictions. And I believe that's where some of the mechanistic modeling can help. So a couple of slides on E-Therapeutics and where our computational issues lie. So what we do is we model disease cells as networks. There's a couple of papers I put up here just to say that this isn't a particularly out there view. Basically, disease cell, any cell really, their function arises because a collection of molecules within that cell are talking to each other, and you can't really assign function to individual proteins, but rather to collections of proteins. And people in the last decade or so have been looking at this in a disease context, and there's a couple of papers here from slightly different angles that do that. So in cancer, there's the idea of a dysregulated network module. So as errors occur from wherever within a cell, certain functions become dysregulated due to various different types of mutations, but other potential insults into the system. But it's the change in the network that's key, that gives rise to properties of cancer cells. Another interesting one comes from the Huntington's field. So this is a curious one. You know, Huntington's is caused by problems in one gene, one protein. However, the whole field has moved, rather than to looking at manipulating that one protein or gene, to actually looking at what happens after that gene's been mutated. The network in the cell rewires effectively. You end up with a different cellular function than you would in a normal state, and that's what gives rise to the pathologies of Huntington's patients. And so there's not a lot of point in treating the trigger. It's too late by the time you're treating the trigger. You've got to treat where you've gone to, and that involves a network. One of the biggest properties of biological networks comes back to this work that was done, and other networks I should say as well, transport networks. The internet had the similar properties. It was done in the early 2000s, was that they're robust to random failures. So if you take a network, a biological network in this case, and you randomly remove nodes, and you measure some measure of impact, I've looked at the differential of the average shortest path across the whole network here, not a lot happens. You can remove quite a lot of nodes and have very little effect on the network. However, if you choose your nodes, you can have a much bigger effect. So biological networks are robust to random failure, but they are susceptible to targeted attack. So the approach E-Therapeutics is taking is to build networks of the disease processes we want to disrupt with a drug, and then look for collections of proteins that will have these sorts of effects on the network, whereas the vast majority of things, any random compound will have these sorts of effects. So what are our upcoming issues, I guess, with where we need new computational approaches going forward? Biological data is horribly noisy and incomplete. We would like, for a lot of our things, to move towards a more statistical analysis to take account of some of those issues. However, that would potentially raise our computational overhead by 3-4 orders of magnitude. So currently it's not feasible, basically, for us. We've been working with Andrei Markov at Newcastle and in collaboration with the Poets on an earlier sort of smaller version we've called Fantasy, that effectively takes our biological networks and embeds them in hardware. The talk yesterday on the Poets mentioned, the idea of going back to the original maths, thinking about what the maths does, and sort of re-implementing those algorithms on the FPGA hardware, and that's exactly what we did here. What Andrei did here. I didn't really have much to do with it, and we got about 3 to 4 orders of magnitude improvement in speed over our current distributed grid system. So the guys in my team now are actually starting to think of analysis that are currently not possible. They're basically sort of rethinking, what's feasible in this context. And hopefully, as we go to the biggest system of Poets, that will even get better. One of the simplifications we take, our networks, we call them protein networks. They're not. They're actually measured at the gene level due to experimental limitations. It's a pretty massive simplification. Data is now starting to come out at the protein level. However, we've got about 20,000 genes. There's probably over 1 million different proteins. So the order of magnitude of problems has, massively increased once we move to this level. This data is probably going to be available in the next decade. Dynamics. We model everything as a static network at the moment. That is certainly not the case. Signal transduction, for example, is a drug applying to there, basically operates as a dynamic process running on top of those networks. Another level of time, networks, I've mentioned this already, rewire as a result of perturbations continuously due to environmental triggers, cancer development, but also as a result of drug perturbation. There is some early work that's going on now as in what's called temporal combinations. You apply a drug at this point in time, the cancer cells, for example, rewire into another state. You know what that state is. You can apply a second drug that will attack the second state, potentially the third state. That's, again, very early academic work at the moment. And we model things right now at the cell. There's much higher levels, intracellular interactions and higher system level interactions. So that's enough of what we do. So personalized medicine. So the current state of the art of personalized medicine is sort of illustrated here with the breast cancer case. So the idea is, in cancer at least, you do a biopsy, you get molecular profiling of the individual, and you get a specific treatment of the individual. So breast cancer, HER2 positive. HER2 is one protein receptor. Does the individual have that receptor? If so, then these drugs might work. Another example, if they're HER2 negative, they don't have the HER2 protein, but they have one or two hormone receptor proteins. One of these two drugs might work. That's great. It actually has revolutionized the treatment of breast cancer in certain cases. However, there are some issues. So only 20% of people have HER2 positive breast cancer. The overall response rate to one of the best drugs for HER2 positives is 26%. So, what's that? Quarter of 10, that's 5% now we're down to treating. Triple negative breast cancer, which you've probably heard about, basically means you're hormone receptor negative and HER2 negative. There's actually no targeted treatment at all at the moment. So while personalised medicine is certainly making advances, there's a long way to go before we've solved all of the issues. So one of the issues here, this diagnostic test is based on 3 proteins. As I mentioned, we have 22,000 potential. There's a PAM50 test, which is based on 50 genes, but currently it's prognostic only, and it's not used for treatment. So the bottom line is that cancer, even in one tissue, is caused by multiple mechanisms that are different in each individual. And this heterogeneity is the leading cause of failure due to in drug discovery, or one of the leading causes of failure. So, if we can improve the predictive possibilities here, as I mentioned out here, of basically identifying the unique mechanism that each individual has and then looking for drugs that will treat that mechanism, we can improve both the treatment of individuals and the productivity of the industry. So what do we need? So biopsy markers, I'm rushing a little bit here, I'm running out of time, but biopsy markers, that's basically sequencing of the individual's genome, potentially other system-wide measurements from diseased tissue. Now that's limited to certain tissues. You don't want to go and take a biopsy of the brain, for example. So it's not particularly suitable in most cases for early detection. However, blood markers are. That's easy to take. However, there's obvious issues. But there's a lot of work going on here of sort of early blood marker detection. And the one thing I wanted to bring up is Theranos. I don't know if anyone's heard of this. It's an American company that developed a testing model that could work on very small amounts of blood. And in fact, they got a deal where they had one of their machines in every Walgreens, that's Boots basically in the US, to allow direct-to-consumer blood testing. Problem was, it was a fake. A lot of money was pumped into this, and the science never worked properly, and they basically lied to their investors, and the CEO is now up for wire fraud in the US. However, the reason I put it up there, it illustrates there is an actual massive need. If they could get this technology to work, and there is actually evidence that the technology underneath it probably could work, given if someone didn't bullshit as much and go down the route of lying to their investors, it might well have worked. Unfortunately, that whole field has probably now been put back, a long way because of that. And then the other interesting biomarker development is in certain situations is sort of digital and other biomarkers. So Parkinson's is actually for both of these. Drug companies now have developed apps that they're giving to Parkinson's patients during trials, so they can get continuous monitoring on their phone of their gait, etc., as they walk. So now you can look at continuous improvement rather than the traditional, which is go and see your doctor once a year. And then you've got the variances of, you weren't feeling very awake that day, you went to see your doctor, etc. So they're moving to a more continuous monitoring of potential Parkinson's patients on trials. Obviously, that could potentially be also then used for early diagnosis. It isn't right now for ethical reasons because there's no way to treat Parkinson's, so why detect it earlier? The smell one's a curious one. This was a lady whose husband had Parkinson's disease, and they figured out actually she could detect the change in smell of people with Parkinson's disease. They gave her a double-blinded test, and she predicted I think she got all of the current people with Parkinson's correct based on smelling their T-shirts or whatever. She got 2 false positives. She predicted these 2 people had Parkinson's who didn't, and they actually developed Parkinson's a few months later. So people are actually starting to look at how the hell can she do that, and can it be turned into a diagnostic? So there's other sort of digital and non-digital biomarkers are actually being looked at as well, and all of these are critical for both personalised medicine and early detection. One of the issues with personalised medicine is development costs. If every disease becomes only applicable to a small number of patients, then not enough patients are going to buy the drug to actually make it a viable business model. However, the flip side of that is if you can predict this, your clinical trials become much smaller and much cheaper. So I think the economics actually will potentially change in the future as well. Combinatorial computational contributions. A lot of these are inverse problems. We have a lot of measurements that come from wherever, biopsies, blood, and we need to infer the state of the disease system. Given the disease system, we want to make predictions on how to move that disease system into a healthy state. And I think computational approaches, E-Therapeutics has taken some of the early baby steps in that, I think has a lot to add in the coming years. The last slide I have, if I've got a couple more minutes, sorry, I've gone over. I'm not going to read through this, but there's one of the biggest issues that we have in the drug industry is our translatability of preclinical models, and this is particularly the case. I've come back to the dementia I mentioned earlier in Alzheimer's. Basically, the models we use for Alzheimer's don't predict what happens in humans. They just don't. And it's an issue in other drug diseases as well. So, I talked to my colleagues at work about this, and they said I was crazy, that potentially in some of these areas that preclinical animal models can be complemented or potentially replaced by computer models. I was thinking of simulations when I wrote this. We heard of the neuromorphic stuff yesterday, of the potential for novel architectures to actually start, recapitulating real brain circuits in hardware. If we can get to the point where they actively can mirror what's going on in humans, they've got to do better than the current animal models do for some of these neuroscience diseases. I did some brief looking around on this. There's actually been a relatively decent amount of progress in Parkinson's disease on this, from the simulation side, but very little in the Alzheimer's world. And epilepsy is the other one where I believe that some progress has been made. And this is what it needs to do. I'm not going to run over this, and I'll leave it there. Thanks.
Urgent questions. We are right next to coffee, so the other alternative is catch Jonny a coffee.
Yeah.
But if there's anything anybody wants to do now, speak up.